Okay. So for today, I want to give you a brief overview of my vegetable breeding program. I work in two crops, spinach. I'm going to give you a very brief overview of one specific project on seed for grain, and also tell you a little bit about my tomato program. For those of you that are not familiar, Weslaco is located at the Rio Grande Valley in South Texas. This is one of the vegetable producing areas in the state. Then at the end, well, most of the time is going to be about this project that I have been working the past 3-4 years on firmness and shelf life in tomatoes. So in general, my research program is very diverse in terms that I work in applied research on developing cultivar breeding lines and fundamental research on molecular biology and functional genomics, especially on plant-insect interaction. Also work a little bit on production system in terms of evaluating cultivars during the off-season using protected structures. So at the end, what I want is all these areas overlap and help me to develop better cultivar for Texas. So for my breeding program in spinach, the main target of the spinach program is to develop spinach varieties that are resistant to diseases, which are the main problem for production in Texas, which are white rust and anthracnose. For this, I have been working for the past 6 years. We were very lucky to receive a SCRI in 2017, and then we renewed it this year, so we got it again. And this is a collaboration with the breeder at University of Arkansas. So several aspects that I work on that, especially what we wanted to do is to identify new sources of resistance and also develop molecular tools for improving selection. So in my program, I work on molecular breeding, white association for white rust, anthracnose. Also work on different quality aspects like vitamin C content and also nutritional content in the seed. Working recently on genomic selection in white rust, and also working a lot on high throughput phenotyping using drones to help me screen breeding lines for yield and plant growth parameters. Also have been working on water use efficiency using drones. So I have several publications on this. If you are interested, you can please contact me, or you can look up in my website. I have the link for the publication and summaries of my research work. So recently, also have been working on this out-of-the-box project on spinach seed for grain consumption. This was a project that was funded by the Texas Department of Agriculture. I just want to mention very briefly, but I think it's something that I really feel excited about it because I like to work on these out-of-the-box projects. So as I mentioned before, this project was funded by the Department of Agriculture, the Texas Department of Agriculture, and is to try to evaluate the feasibility of producing spinach seeds for grain consumption. The main idea here is to be able to have a double-purpose crop in the sense that we can harvest the leaves and then let the plant to bolt and then harvest the grain. The idea here is to have an additional source of income for producers. As you know, with the leaves in spinach, but other parts of the plant are also edible, like the stems or crowns, and also the roots are edible. But not much attention has been given to the grain, mostly because what we call seed actually are the fruits, have a very hard fruit coat, so it's not very something that you think that is edible. But if we remove the fruit coat, we can get the seed. So spinach is in the same family as amaranth and quinoa, and as you know, amaranth and quinoa are high value grains, and they have a high nutritional value. So one of the first questions that I wanted to answer is that what is the nutritional value in spinach grain as compared to amaranth and quinoa? So I did several evaluations in terms of mineral content and also amino acid content, and we compared it with amaranth and quinoa. So briefly, one of the major results from this was that this protein digestibility test. So essentially, protein digestibility is a method for evaluating the quality of a protein based on both the amino acid requirements of humans and their ability to digest it. So what I was very surprised was that as compared to amaranth and quinoa, spinach is twice as much, has a more digestibility score as compared to this highly valued. So I think there is in the terms of nutritional quality, there is a potential for spinach grain. We also did, well, the most important to see if there is a economic incentive to do this. So we proposed to do this, that harvest the leaves and then leave the plants on the field, fertilize one more time and water a couple more times. That will result in a cost of $300 more per acre for a producer. So what we did was a sensitivity analysis, try to see what price we have to sell the spinach grain and how much yield do we have to produce in order to break even if we do this. In other words, how much do we have to sell and how much do we have to produce in order to recover those $300 extra? So what we found is that we need to sell the spinach grain at 20 cents per pound and produce around 1,000 pounds per acre. So we don't have a reference price for spinach because it's not commercialized as a grain. But if we compare something with amaranth, that is 40 cents per pound for normal market, or organic is 50 cents per pound. So I think we have very good chances to be successful if there is a market for spinach grain. So also in terms of yield, we will have to produce around 1,000 pounds per acre. So most of the spinach land have been bred for low bolting. So the average yield is we can break even, but we also screen for diversity in terms of yield in the field. And we can produce up to 8,000 pounds per acre. So that will result in about $3,000 to $4,000 per acre just by investing 300 more if you leave the plants on the field. So I think there is some potential in here. We have been working also to see if we can improve yield in terms of grain, but also nutritional quality. There is a lot of diversity. So my hope is that in the future, we can have something like that. Like we have in amaranth and quinoa, there are several products that people are willing to buy. So like bread, like quinoa that is mixed with rice, granola bars. So I think this is something that can potentially be an additional source for producers. And it has a good quality in terms of compared with other grains like amaranth and quinoa. It's like, have you tasted quinoa? Or it doesn't have actually a strong taste. It tastes like the condiment that you put. It's like rice. It's not a. So for the tomato program, so the idea is to develop cultivars adapted to Texas conditions. So I have been working on conventional tomatoes, but also in these specialty ones with different colors, textures, and flavor, because they get a premium price. So if we want to be competitive, we need to get in these niche markets. Also, I have been working in these cherry type tomatoes, F1 hybrids. I have been testing for the last 3 years, and I think we are ready to release them next year. With the final evaluation this year. So mainly we are targeting these ones to produce during the off season in high tunnels. So we have seen that we can get very good yield and also harvest during the time of the year where the tomato prices are higher. So there is a lot of potential also on this. So for my breeding, I work on mainly On disease-resistant, tomato yellow leaf spot virus is one of the main problems that we have there in Weslaco, in the Rio Grande Valley. Working on molecular breeding, quality, market-assisted selection. So one of the things that I'm doing is to stack different tomato yellow leaf spot virus-resistant genes. And we have observed that by stacking at least 2, it provides very good levels of resistance there in South Texas. I have been working a lot on high throughput phenotyping as well in tomato. One of the main things that it costs a lot of money is harvest the tomatoes and to estimate yield. So we can, now we have been working on several aspects on this, but we can have a very good yield prediction using plant growth parameters. We are around a 70% accuracy, which is good, but actually we are not interested to be very precise in trying to determine how many pounds per plant or something like that. We are interested more in ranking or lines. So in terms of ranking, is almost 95% accurate. So I think this is a good potential for using this and not having to harvest the tomatoes on the field. Also, I'm using high throughput phenotyping for evaluating for disease. So I think this is very important because normally what we do is go to the field and do visual evaluations for evaluating resistance, but this may be more accurate. As I mentioned before, I work on high tunnel production during the off-season. And a lot of work also, I'm trying to work on improving tomato quality traits, especially firmness, post-harvest shelf life. And for this, actually, this year also I received another grant that I have been working this year. And this is actually the project that I want to share with you today. So the title of my presentation is Tomato Fruit Firmness, Breeding for Long Shelf Life while Conserving Quality is Now Possible. I always like to start my presentation with this slide. So this reminds me that for a good tomato cultivar, I have to have in consideration different aspects. So for example, for a producer, a good cultivar is one that has resistance to pests and diseases, tolerance to abiotic stresses, and of course, a high yield. Retailers also are very interested in shelf life and constant supply. And the consumers are very interested in appearance and firmness. And very often now they are more demanding better taste and high nutritional quality. So I think this is a good opportunity in the tomato industry. So I want to talk about tomato quality. Unfortunately, there is a general consensus that most commercially produced tomatoes have lost their flavor and nutritional quality. So if you go to the supermarket, there is the tomatoes look very nice. They are big, red, but usually don't have a good flavor. And that results in reduction in sales in the supermarkets and consumption. So actually, some years ago, we were approached by HEB and tell us about this, that they are concerned in terms of that tomato quality is not good and people are buying less and less tomatoes. Another thing is consumers repeatedly request for tomato that is vine right flavor, aroma, and texture, and are willing to pay a premium for locally produced selections. Here I have a picture I think I took in San Antonio a couple of months ago. So these are specialty tomatoes ripe on the vine. So these are $2.50 per pound as compared to $1.20 that is usually for regular tomatoes. And heirloom tomatoes are almost $5 per pound. So there is a good potential for producing these tomatoes with better flavor and nutritional content, and consumers are willing to pay more for them. So one of the things is the perception that there is there is a declining in nutritional value, quality, and flavor. But looking into the literature, there is a lot of evidence of that. For example, Davis in 2009 published that the concentration of some nutrients have declined in the last 50 to 100 years. One of the causes of that he attributed to the what he called the dilution effect. Essentially, the fruit and vegetables that we have now are larger, they have more water content, but in terms of nutritional quality is lower. Also, White and Broadley did some analysis on 26 vegetables. So he compared old versus new varieties, and he created this ratio between in terms of mineral content. And as you can see, all the ratios are lower than 1, meaning that the new varieties have lower nutritional content as compared to the old varieties. Additionally to that, another line of evidence is the use of uniform gene. So as you know, we all like tomatoes that look very uniform in terms of very red color. But in nature, tomato doesn't ripen that way. It has what we call the green shoulder. And this was a gene that was found, I think, during the 50s that make the tomato ripe uniform. It encodes for a golden 2 light transcription factor that regulates fruit chloroplast development. The problem with that is that the tomato ripe very uniform, but there is a decline in about 30% in sugar content and nutritional quality and flavor. Another factor that causes a decline in nutritional value, quality, and flavor, and I think is very important, is the harvest and post-harvest both practices and also breeding for this. So for this presentation, I would like to define in a very simplified way what post-harvest shelf life will be, and I think this is an agreement for most breeders, is that the ability of the tomatoes to keep a firm texture from the point that we harvest them, transportation, the shelf life in the supermarket, all the way to the point that we consume them. So usually this process may take 2 to 3 weeks, 21 days. So this is a large process. So there is a lot of time from harvesting to consumption. So the tomatoes need to be firm during that time. Usually the first thing that people do when they go to the supermarket to buy tomatoes, the first thing is to touch them to see if they are firm. The ones that are soft, usually people don't buy them. So there are several approaches that can be used for extending post-harvest shelf life. The one is a cultural practice, harvest green. So that allows the tomato ripe during all this process. However, as we know, vine-ripe tomatoes taste a lot better. There is a reduction in flavor and nutritional content when we harvest green. So we have to trade long post-harvest shelf life for nutritional quality. Another approach will be, this is a breeding approach, delayed ripening or the softening process by, for example, blocking ethylene. There are some genes that have been utilized successfully, the ripening inhibitor and the non-ripening. So here I have a picture of this. This is a homozygous ring mutant. So this is on day 1. After 50 days, it never ripe. This is the wild-type parent having 2 functional copies of the gene. After 49 days, it's completely rotten. So what breeders have done is to produce heterozygous, well, F1 hybrids that have intermediate levels. However, as you can see here, in the case of lycopene, also the F1 hybrid has half of the nutritional content as compared to the homozygous functional copy wild-type parent. So yes, there is a extension of shelf life, but again, a reduction in quality. Also, another approach is to target cell wall enzymes, pectin lyase and polyglycolinase. Those are enzymes that are involved in the softening project. By blocking them, we can extend shelf life. However, as you know, for example, in the case of the flavor saver was one of the first transgenic tomatoes, genetically modified tomatoes, sorry, was not very successful. So there is different approaches. Actually, we have one student here working on this project, trying to modulate the how we can delay the ripening softening. So we can, again, try to improve post-harvest shelf life without having a huge deleterious effect on quality. So another way that we can extend post-harvest shelf life is to try to identify QTLs for firmness. This has been done before using wild-type relative tomatoes. However, the level of success is very minimal. They haven't actually extended very significantly the post-harvest shelf life. In addition to that, we know that utilizing wild relatives have the problem of bringing undesirable traits also. So again, we can improve a little bit, but it has some negative effects. So it's clear here that one major breeding goal in tomato is to deliver improved flavor and nutritional content in the context of high yield and long post-harvest shelf life. So ideally, what I would like to do is to develop a tomato that we can harvest vine ripe, so have good flavor and nutritional quality, and that can last those 21 days that in the shelf being very firm. However, up to this point, with the project that we have been attempted, that I mentioned before, this has not been possible. So we have to either select for good quality or for long shelf life, not for both of them. So one of the first things that I tried to do in order to improve firmness in tomato was to evaluate my breeding population in terms of quality traits, including firmness. So we evaluated 305 Texas A&M breeding lines for 2 years. We were able to do a wide association study and identify several QTLs for firmness. However, all the effect of those QTLs were very minor. So that makes it very difficult to improve firmness. However, when looking into the distribution of firmness in the population, we observed this line that was very, very firm. So for now, I refer to firmness as a kilogram force, which is the force needed to puncture the tomato with a 3 millimeter puncture device. Essentially, measure the force needed to puncture the tomato. So this tomato line, 157, was exceptionally firm, way firmer as compared to the mean of the population. Just for your reference, tomatoes in the supermarket when are very firm are around 0.8 kilogram force. So this was way more firmer. So this tomato line has a very high compression peak, 1.571 kilogram force. So I would say it's even harder than a green tomato. Has a high dry matter content, around 10%. This is as compared to tomatoes in the supermarket that have around 5%, so a lot less water as compared to tomatoes in the supermarket. Another very important characteristic, is, hopefully you can see here, is that it doesn't have locule gel. The gel that surrounds the seeds. So this can be a good characteristic because a lot of people don't like when you slice it and put it in a sandwich that the tomato leaks water because that makes your sandwich soggy. So this is something that may be a potential application of that. In terms of flavor component, bricks, and acidity, it has a very good bricks around 0.7. For reference, the tomatoes in the supermarket are around 3.5. So they are way better tasting as compared to the tomatoes in the supermarket. So having in consideration that has a higher dry matter content, it has no locule gel that doesn't leak. So I think one of the natural ideas to test is that results in extended post-harvest shelf life. So we did some preliminary evaluations. So we harvested fully ripe tomatoes, and we evaluate firmness up to 1 week, up to 9 days. using our tomato line 157 and some F1 hybrid developed at Texas A&M, Tam Ho Tai, and also commercial hybrid. And as you can see, our tomato line keep firmness for 1 week very well. We extend the trial to all the way to 21 days because this is the time that we consider that tomato need to be firm. So again, we we harvest the tomatoes completely ripe, red ripe, and evaluate firmness at room temperature and refrigerated for 21 days. And as you can see here, after 21 days, even when we kept the tomatoes at room temperature, the firmness was equivalent to a tomato, a commercial tomato that what you can find in the supermarket. So it's telling us that we can extend post-harvest shelf life with these tomatoes, because they are still firm even after 21 days when they were kept at room temperature. So I think with that, we have the hypothesis that we can propose another approach to extend post-harvest shelf life that will be to select for high dry matter content. In this case, taking advantage of the high dry matter content in this tomato line and also to the lack of locule gel. I do always this analogy as compared to sun-dried tomatoes. If you remove the water in the sun-dried tomatoes, that's why they last longer. And because they have less water, they have more intense flavor. So potentially we can improve both post-harvest shelf life and also flavor in tomato. So we decided to test that. So what we did was to cross a tomato line that has locule gel and it's very soft with the line 157 and cross it to produce an F1 hybrid. So just for the purpose of this presentation, the presence of the locule gel I'm going to label as a double capital G, the absence of locule gel as a lowercase g, and the heterozygote, of course, 1 capital and 1 lowercase g. So we harvested tomatoes at different stages, mature green, breaker, plus 3 days. So breaker is the stage where the tomatoes start to turn red. So we tagged them and waited 3 days, 9 days, and 15 days after breaker stage. And we evaluated for post-harvest shelf life at the time of harvesting, 7 days later, and 14 days later. So very interesting result that we obtained. The first one that was that the F1 tomatoes, when crossing a tomato that has the locule gel with a tomato that doesn't, with the line that doesn't have the locule gel, all the progeny results in tomatoes with locule gel. In addition to that, when we observe the firmness as compared to the parental line that has locule gel, that is soft as compared to the line 157 that is very firm, the F1 has intermediate values. So since the F1 has locule gel, we hypothesize that yes, the locule gel contributes to firmness, but in addition to that, there is a potential that other factors in the pericarp contribute to fruit firmness in the F1. So it's not only the lack of locule gel, but also the other factors in the pericarp. Also, I think this is very good because we can produce tomatoes either with or without locule gel that are firmer. So that will satisfy a broad range of consumer preferences. Another very important thing that we wanted to evaluate is at what stage we can harvest these tomatoes and still be very firm. So as I mentioned before, we collected a mature green, breaker +3, all the way to breaker +15. And as you can see here, in the tomatoes that have locule gel, there is a quick decline after a breaker +3 as compared to mature green. But in the case of our tomato line 157, it seems that firmness still is kept even after 15 days. If you compare to the soft parental line, the level of firmness is very good. It's above a 1. That is even better than the tomatoes in the supermarket. So what this is telling us is that we can wait more to harvest the tomatoes and wait until they are fully ripe in order to harvest them, and still they are going to be very firm. So another aspect I didn't mention before in terms of evaluating post-harvest shelf life is the moisture loss. As we know, upon harvesting, the tomatoes start to lose moisture. So we evaluated also that after harvesting, the day of harvesting, 7 days later, and 14 days later. So the very interesting thing in here, as you can see here in the tomato parental line that has the locule gel and in the F1, after 1 week, 7 days, there is a 4% loss, similar with the F1 hybrid. But in the tomato line 157, it's only a 2% loss weight. And after 14 days, it's an additional 1%. So after 14 days, it's around 3% loss of moisture, as compared to the tomatoes with locule gel that have around 6% moisture loss. So this is telling us again that we can harvest later and still the tomatoes can be kept, in this case, at room temperature and conserve the quality after 14 days. So another aspect that I have been working on is on firmness trait inheritance. So we cross with different lines that are soft and have locule gel with our tomato line 157. We develop F1 hybrids. Then we look into segregation in the F2, and also we did some back crosses to corroborate inheritance. As I mentioned before, it doesn't matter the background that we cross the tomato line. So all the progeny results in all the F1 hybrid results in tomatoes with locule gel. This is a good indicator of a single recessive gene. So we went ahead and we screened the F2. And we obtained the expected ratio. So the expected ratio for a single recessive gene is, in this case, will be 1 with no gel to 3 with no gel, and we almost got a perfect ratio. So we were expecting 25% of the tomatoes with no gel and 75% of the tomatoes with gel, and this was essentially what we obtained. So at this point, we think that there is the lack of locule gel trait is controlled by a single recessive gene. Even though that I think I was pretty sure at that point, I'll always like to corroborate that. So I backcrossed the F1 with a parental line that has the locule gel. So in here, all the backcross one is expected to have 100% tomatoes with locule gel. And out of 165 that we evaluated, all 165 were having the locule gel. So that's a pretty good indication that a single recessive gene is controlling. We also backcross it with the parental line lacking the locule gel. So in this case, the F1 is heterozygous and the parental line is homozygous. So we were expecting 50 and 50% of tomatoes with no or with locule gel. And we got a very close ratio. So at this point, I'm very sure that the lack of locule gel trait is controlled by a single recessive gene in the tomato line 157. Another aspect that I was very interested on evaluating is trait heterozygosity. The idea here is that if we can use the line 157 as a parental line to develop hybrids, would improve nutritional quality, flavor, and also firmness and post-harvest shelf life. So here what I have is the value for Brix and acidity. Those are components of flavor in tomato. So here I have the value of the line 157. I cross with different soft parental lines. So here I only have the average of the soft parental lines and the average of the F1 hybrids. So as you can see, there is an increase of 11% as compared of the soft parental line and an intermediate level between the two parental lines. More important, as compared to commercial check, is significantly higher as compared to commercial check. Similar story with the titratable acidity. There is an increase of 10% as compared to the parental line, the soft parental line, and is significantly higher than average commercial shade. So our line can be used to improve, in short words, our line can be used to improve flavor and develop F1 hybrid with higher solid content and titratable acidity. We also evaluated Firmness, very similar results. So there is a 30% increase in the F1s as compared to the soft parental line. Again, significantly higher than the average commercial check. A 50% increase in dry weight as compared to the soft parental line. Again, significantly higher than the commercial check. So together, we can tell at this point that there is a possibility to improve both, in this case, flavor and potentially nutritional quality, and at the same time improve firmness, and of course post-harvest shelf life. So we don't have to trade between or choosing quality or choosing a post-harvest shelf life. Again, the only thing that remained to be sure that this is a good parental line is to see if that the hybrid have higher yield, because as I mentioned before, we want to improve quality and post-harvest shelf life, but in the context of high yield. If we don't have tomatoes that produce a high yield, so it's pointless. So the results indicate that in average, the tomatoes produce a lot of tomatoes comparable to the average commercial check. So again, yes, we can produce tomatoes with better flavor, with enhanced post-harvest shelf life that also yield good. So very excited for the results. Another thing that I've been working on is on mapping Firmness traits. Mapping the Firmness traits. So we develop a biparental population sequencing, and also map it using Windows QTL cartographer. One of the very interesting things that we found in the correlations in the F2 is that the presence of the gel in the tomato negatively correlate with pericarp firmness, mesocarp firmness, Brix, and acidity. In other words, if tomatoes without a locule gel result in more firmer tomatoes and higher Brix and higher acidity. So this is something that we're very excited about. For the mapping, we identified that the recessive gene controlling the lack of locule gel is localized in chromosome 10, while the other QTLs that contribute to firmness in the pericarp are localized in chromosome 8, 9, and also 10. Just as a summary, we are trying right now to identify putative genes controlling the firmness. For example, for locule gel, we were able to identify 5 genes near to the link marker, but looking to the expression, only 1 is expressed in the fruit, and we haven't found any difference in expression in the tomato line and in the wild-type tomatoes. So at this point, I think we need to improve our mapping, and that's what we're trying to do. In terms of pericarp firmness and mesocarp firmness, for pericarp firmness, we only identify 1 QTL, and we identify around 500 genes around it. And I think, again, we need to find map to narrow down the potential candidate. Similar for the QTL for mesocarp firmness in the chromosome 8, 9, and 10. So also looking into the literature, actually in 2021, this paper was published. So I was a little bit surprised because the phenotype is very similar of the tomato line 157. So they call it the all fresh fruit. What they have is a tomato that also doesn't produce locule gel. But in this case, the phenotype is different in terms that it doesn't produce locule gel, but it produces solidified gel. In the case of the tomato 150 line that I have, it doesn't produce this solidified gel. And also what we observing here is that there is no presence of anything. The seeds are attached to the tomato without any surrounding solidified gel. So maybe something similar, but the phenotype is clearly different. Anyway, I decided to double corroborate that we don't have the same mutation. So fortunately, they were able to map the gene. Actually, they map it on chromosome 6, while we have mapped it on chromosome 10, so we don't think that is the same. Also, they know that the gene that result in all flesh fruit mutation is the MVP3, that it calls for a mailbox transcription factor. Actually, they developed some markers, and they know there is a deletion of 400 base pairs in the all flesh fruit. We don't have that deletion in 157. So I think there is maybe potentially in the same pathway, but not the same mutation. And the phenotype that they produce is definitely different. But another thing that I was interested in this paper was that they found that the change in locule gel was as a result of a mailbox transcription factor. So mailbox transcription factor have been related to fruit development. So one idea that I had was to check if the transcription factor in chromosome 10. As you can see in the all fresh fruit, in chromosome 6, this MVP3 is highly expressed in the fruit, and is highly expressed in the locular tissue, here in red in the tomato. So my idea was to identify a potential mailbox that is highly expressed in chromosome 10 and that is highly expressed on the fruits in chromosome 10. So I look on all the already known transcription factor in chromosome 10, mailbox transcription factor in chromosome 10, but none of them has significant expression in the fruit tissue. So we don't think that is a transcription factor, mailbox transcription factor. Only one of them have a little bit of expression. We try to see the expression in the locular tissue, but it's very not that much. So at this point, either it's not a mailbox transcription factor in our line 157, or we haven't identified it yet. Again, also this gene was very far from the site from the loci that we identified the potential place for the lack of local gel gene in 157. So as a summary, line 157 can be used as a parental line for hybrid cultivar development. It increases fruit firmness, dry matter, bricks, and acidity, and of course, in the context of a good yield. This is very important for me. Another lack of local gel trait is controlled by a single recessive gene. Also, we identified that additional QTLs in the pericarp improve fruit firmness independently of the local gel. And again, as the title of my presentation is, we think that it's possible to breed for long shelf life and quality in the context of high yield. So what I'm going to be working on, we are going to try to find map and identify the causative genes for lack of local gel and the QTLs. I didn't talk a lot about determining the effect of local gel and fruit pericarp firmness on nutritional content in tomato. For this, I'm working with Binu Patil, so he's going to help on evaluating nutritional quality in this tomato. So one of the things that we believe is that because it has less water content, so nutritional, the concentration of nutrients is going to be higher. At the same time, by lacking the local gel, may be a shift in nutritional composition. So that's something that we want to find out. And then we wanted to determine the effect of local gel and pericarp fruit firmness on tomato ripening and shelf life. So we hypothesize that this tomato line doesn't have any effect on ethylene emission because it ripe very natural. So usually the mutations in ethylene result in tomato that never ripe. But still, we want to double check, corroborate. One of the things that we know is that ethylene production in the tomato start in the locule gel. So we want to see if that may be what is causing that the tomato has a longer shelf life because it's delaying a little bit, but not blocking it. So that's a good strategy also for improving post-harvest shelf life. Finally, another thing that I'm very interested in I'm working on is on yellow shoulder. Yellow shoulder is a tomato, is a disorder, a physiological disorder. It's not a delay or problem with ripening, but it's a physiological disorder that results in the fail to develop red pigment. And the problem with this is, one is in terms of appearance, it doesn't look nice. The other thing is it affects flavor and also nutritional quality. If you see on the slide a tomato that you buy in the supermarket, all of the tomatoes have this problem. And as you can see, line 157 is very uniform, doesn't have barely a little bit of this. So I think we can improve also appearance in tomato using this breeding line. So actually, we have started to try to map this for QTLs. We have been doing a visual assessment of the degree of yellow shoulder. So far, we haven't been able to obtain significant signals on that, but we are working on that. Hopefully, we can have more information soon. With that, I would like to thank all the people that has contributed in this project. Dr. Jana Rameneni, he's a recent scientist in my lab. He's working on the characterization of the tomato line. Amo Duragbeni, he's a PhD student that joined my lab this semester. He's going to be working in this project. Adam Munoz and Aizar Garza, both of them are research associates in my lab. They are the responsible and they are the most valuable people in my lab because they are the ones that keep the plants and in the field and in the greenhouse, do the crosses. So I'm very thankful with them. Also former member that work on this project, Debbie Kandel, Faridul Islam, and also Henry Awika that have helped on this project. Of course, collaborators, Kevin Crosby, Kranti Mandari, and Bimu that are working in this project. Also, our funding source, the USDA, that allow us to continue with this project, and especially to the Texas A&M Marina Research and the Vegetable Fruit Improvement Center. So all the preliminary data that we collected was used to submit this grant that fortunately was awarded this year. So very thankful for that. With that, I can take any question that you may have. Yeah. Actually, it's an inbred. So she's asking about if we commercialize the line 157. At this point, we don't want to commercialize it as a final tomato product. We can utilize it as a parental line to improve. But yes, definitely this is an inbred line, so yield is lower as compared to the F1 hybrid. That's the main target for cultivar development. Yes.
Is it indeterminate or determinate?
It's a determinate line, the tomato, the 157. Actually, yeah, that's another possibility. One of the things actually I'm thinking about is that yellow shoulder is very important in the processing industry. So tomatoes that have the yellow shoulder cannot be sold as a diced tomato. So they have to be sold as for making ketchup, and the producer receive a lot less. So, for example, if we can improve yellow shoulder tolerance using this line, that will be something that have a very good impact in the industry.
The yield is
yeah.
Color,
definitely does. Yes.
See a correlation between firmness and difficult?
Not really. This is no, we don't have any problems in terms of. One of the ideas that we have is that let the tomatoes ripe on the vine, so they have a better flavor and nutritional content. Of course, leaving the tomatoes on the plant may result a risk in terms of pest diseases or problem with a freeze or something like that. But yeah. Yes, that's something that I'm very interested. I usually give to the people to try the tomatoes, and so far I have received very good responses. Actually, we have several undergraduate students going to visit the lab, and also high school students going to the center. So every time that I show them the tomato, they say that usually they don't like tomato, mostly of the. And one of the reasons they say that they don't like it is that because they leak water and they make the sandwich soggy. So they were very interested on that, and they say they probably eat these tomatoes because it's not that not leaking water. So I think there is a potential of that. In terms of flavor, it's very subjective. But what
You want
not subjective part, we can tell you that in terms of bricks and acidity is even higher. Another thing that I didn't show during my presentation is very interesting. What we have found is that regularly the tomatoes tend to, after harvesting and evaluating post-harvest shelf life, they tend to lower the acidity. But the tomato 157 tends to increase acidity. So this is a very, very interesting thing. At the end, I think in terms of acidity, they after storage, they match. So it's something very, very interesting that we identify.
Hello, Dr. Avila.
Yeah.
Yeah. I have a question. Like, here, like the tomatoes, like when you have almost no locule. So when tomato, like my question is about like at what rate these tomatoes ripen, because most of the like when tomato starts ripening, the first tissues like which produces ethylene to start ripening process. So it comes from locule, but when we have no locule, so how like this tomatoes like ripen, and do they have like what's the rate of their ripening? Is it very slow? Because if it is slow, it's a benefit because like post-harvest handling wise, so that is a plus point. But what happens to the normal ripening of tomatoes? Do they ripen normal?
Yeah, well, the question is if there is if the tomato 157 line has a different rate of ripening. As we can observe, the tomato line ripes normally, but as you can see, the data suggests that it still keep firmness. So one of the things that we are evaluating is that ethylene emission. So we have very preliminary data. That's why I didn't want it to show in here. But actually, we have very interesting results. So I was thinking that this line will have a delay in terms of ethylene production.
Yeah.
But actually, we observed that in the very preliminary, so these are not conclusive yet, there is actually higher emission of ethylene as compared to the other tomatoes. So I was very impressed on that. We are planning to repeat this trial this season, so we will have better understanding on that.
I think I have just one more question.
Yes.
Like like when did you get chance to like measure ascorbic acid levels in tomatoes like in this breed of like a tomato? Because most of the ascorbic acid in tomatoes, they are in locular tissues. So in absence of locular tissue, do the level of ascorbic acid goes down?
Sorry, I didn't understand very well, but in terms of nutritional content, we don't have data yet So that's something that we are evaluating at this time. Yeah, but
No problem.
or we expect that, as I mentioned before, probably we will have a higher concentration, but a different profile in terms of nutritional content. That's what we expect.
Okay, thank you.
Mhm. Mhm.
To develop hybrids, we will always have hybrids that have
Yeah.
getting into conversion programs where you by crossing end up developing versions of very important lines that actually do not have the local gel, but then that way those would be perfect for developing hybrids.
Yeah, actually, yes, we are already. I have crossed this line to several of my elite lines with that purpose, because I think there is some potential to that develop a hybrid that also don't have local gel. Yeah, 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 yeah. Actually, yeah, it's something that we don't need markers for that. It's yeah. Yeah. Yes. Yeah. 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 I'm actually doing that. Yeah. Yeah. Definitely, that's something that I'm doing. I'm back crossing. Actually, I think I already have busy trees on that. Yeah. 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 Right. Yeah. That's not yet. Yeah, that's another possibility. I have been a little bit discouraged because it's far away from the where we identify. But yeah, that's definitely a possibility. Yeah. Yes.
I was kind of wondering with our discussion we had earlier with seed germination.
Yeah.
I know there's a lot of seed germination inhibitors in the locker
Yeah.
gel. So when you remove that, you have issues of seeds germinating in the fruit.
No, actually, we haven't observed anything on that. So what I think, I haven't looked in detail, what is really happening. So this is something that I am really interested. But I don't know if that the membrane is still in there. It's just that the liquid has dried out. So probably all the inhibitors for germination inhibitor are still in there. So we don't know. But in terms of germination, we don't have any problem with germination. Actually, this trait that was published before, the all fresh, they found that they have a reduction in germination. Then they later identified that it's not the MVP3 gene, is another allele that is tightly linked. But yes. Yes. In terms of no, actually, we haven't had any problems with germination or viability. One of the things that I think makes it a lot harder is to collect the seed, because it's like glued together. So when it's not like in regular tomatoes that is very easy to clean. In this case, it's very tight together. So sometimes we have to do it manually. So this is the only one problem that I think I have observed. No, actually, I haven't done that. But let me see if I. So what have been seen that we have tomatoes with high number of locules, with and without a locule gel. So, and another thing, very interesting thing. So let me. Is that maybe there is a correlation. The presence of local gel result in higher gel of shoulder. So by selecting for lower local gel or absence of local gel, we may be able to improve gel of shoulder. So that's actually another observation that we have.